My name is Lars Østergaard and I'm heading the Genes in the Environment strategic program at the John Innes Center in Norwich. The Genes in the Environment program is based on a long tradition at the John Innes Center to try and understand how plants grow, from the very early stages of seed germination to the last stages of seed dispersal, to try and see if we can manipulate plants to be more resilient to changes in the environment, such as those we are experiencing in this period. We go all the way from excellent fundamental science carried out in the lab, out to the field at our field station to do field trials and test how these discoveries they translate into the field. But we also go the other way. We also take discoveries from the field and bring them back and test them under very controlled conditions in the lab. We work on both important crop species such as wheat, brassicas and legumes, but we also work on model species, and these model species are crucial for us to get this fundamental knowledge to then translate into the improvement of some of our most important crops. You know, the year 2015, 2016 was a very bizarre example of what is going on right now, where we had Christmas Day in 2015 was warmer than the 1st of June in 2016. And our crops get very confused about this. So they flower early, they flower too early, and this impacts yield in a very negative way. So we are trying to understand the genetic mechanism that underpins plants' responses to changes in temperature. And by doing this, we will then be able to breed for varieties in our most popular crop species like wheat and brassicas that are then resilient to these changes that we are seeing at the moment.
So one of the examples of work we've been doing here is looking at the influence of climate on oilseed rape. We've noticed that oilseed rape yields are influenced by the autumn and winter temperatures, and we can see this from historical data. So what we've done is we put soil warming cables into plots in the ground at the field station, and then we can compare controlled plots and warmed plots to see how that influences yield. And we found that an October window is particularly important for influencing the yield of oilseed rape the following spring. And it's really exciting because what we can do is take that information then back into the controlled environment rooms that we have at the John Innes Centre and look at the genetic components of what is influencing those changes that we're seeing in oilseed rape yields.
We are experiencing less land available to grow our crops, and this means that we will be able to produce less food for a steeply increasing global population. And there's no doubt that if we don't do anything to address this, we will experience extreme famine at a scale we probably have never experienced before. We can only get there if we really understand the processes that we are dealing with. And we need to take all the methods and all the technologies that are available to us to get there. Computational sciences, including artificial intelligence, machine learning, but also genetics, genome sequencing, to really nail down in very fine details how to make sure that we can feed an increasing global population under those extreme climates that we are facing.